Thanks, Michael. Thanks, ARM, for the opportunity. It's a great meeting once again this year, and thank you all for hanging in there. I know I stand between you and your break. So I'm going to spend a few minutes talking to you about RepliCel Life Sciences, which is a cell therapy company leveraging the cells derived from the hair follicle. So in that sense, it's one of the unique companies in the world because it's one of the only companies that I'm aware of using that as the cell source. Am I responsible for pressing? Oh, yeah, there we go, the big green button. So we're a publicly traded company based in Canada, trading on the TSX Venture and in the OTC in the United States. So I'll make some forward-looking statements. You should do your own homework. So as I said, we're a cell therapy company looking to move into regenerate hair growth, restore damaged skin, and repair chronically injured tendons. So you have the luxury also this afternoon in this session of hearing the only two cell therapy companies that are actually pursuing the treatment of tendinopathies. We've moved 2 of those products into the clinic this year, so we have 3 products in clinical development. And a device, a dermal injector device also that is targeted for CE mark next year. So I'll give you some granularity on each one of those as we walk through the deck. This is essentially a simple schematic of the cell therapy process. The patient is diagnosed. Both of our cell populations are derived from a single suture biopsy from the back of the patient's head. In that biopsy are 20 to 25 growth stage hair follicles from which we've derived 1 of 2 cell populations, depending on the product we're looking to manufacture. The dermal sheath cup cell populations, which you see at the base of that microscopic hair follicle image, is the cell population which we believe can be responsible for the treatment of androgenic alopecia, otherwise known as pattern baldness. The other is a fibroblast cell population, which has nothing to do with hair growth at all, but is responsible for the maintenance of the tissue that comprises the hair follicle. It's a fibroblast in many senses of the word, but it's unique in one respect, and this is the respect with which we intend to leverage it, and that is its high expression of type 1 collagen. So in the published literature in our hands, it's 3 to 5 times more expressive of type 1 collagen than any other fibroblast in the human body, and that's what we expect it to do in the places where we put them. So it's a simple cell expansion play. We expand the cells over 4 to 6, 5 to 8 weeks, depending on the product we're looking to manufacture. Unlike a lot of cell therapy, autologous cell therapy companies, we cryopreserve in the aseptic vial and we ship it back to the clinic for re-injection. And in the case of the scalp and the skin, we have a proprietary injector device for reasons not unlike Ken described in the previous talk. So we believe the fibroblasts are truly a platform technology. If we can treat the tendon in your ankle, we have no reason to believe why we can't treat the tendon in your knee and both the tendons in your elbow, etc. And the dermal sheath cups is a single product focus for the treatment of hair loss. So the other thing I think that's important to keep in mind about RepliCel Life Sciences is we do not aspire to take things commercial. We believe that the most value you create for shareholders is in that early stage discovery to mid-stage clinical development, and then we want to license with sophisticated partners who understand how to bring products into the markets for which our products are being targeted, and we want to also leverage their capital and their infrastructure and their expertise for those late-stage trials. So that's the licensing strategy. The business strategy is to keep things as virtual as possible. We don't invest in bricks and mortar. We contract manufacture. We use contract research and contract people as much as possible to try and focus as much of our capital on R&D and maximizing the assets as possible, including, and I think this is important to understand, including the things which are ancillary around the biologic yourself. And I think we've seen far too many cell therapy companies in the past focus so dependently and myopically on the product, on the cell therapy product, that they perhaps forget to invest or just assume that the problems around manufacturing, cost of production, delivery into the patients will solve themselves if you deliver the right kind of biologic. And we believe, as an asset building company, that the right thing to do in terms of building the value around those biologics themselves are to invest in those things and invest in those things early. So we're investing in the manufacturing, in the packaging, in the delivery, and the other things which build value around your core biologic. So we also have a very sort of simple philosophy of life. There are very many elegant science projects and clinical development projects out there treating very sophisticated diseases. But we have a cell population that we think we understand what it does. We grow more of them. We don't modify them or differentiate them. We put them locally in areas where those cells can perform that very simple function. We don't deliver them systemically, and we depend on those cells to do what we understand them to do. Very simple explanation of the science. The dermal sheath cup cells is the original technology that the company was founded on. It's this cell population that's attacked by the androgen hormone from those people who suffer from androgenic alopecia. There's a direct correlation between the androgen attack on that cell population. As that cell population diminishes, the hair fiber diminishes. What we also know about androgenic alopecia, as you've observed in most of your everyday life, is that most bald people still have hair at the back of their head. And that's because the cell population, the hair follicle and the cell population in that hair follicle are insensitive to the attack of the androgen hormone. And that's why microtransplant surgery works. You can take a follicle at the back of your head, transplant it to the top, and that follicle will continue to grow hair for the rest of your life. But it's a bloody, messy, expensive, highly variable procedure. And so what we're looking to do is capture the cells that we believe are ultimately responsible for that cell cascade that grows hair, simply grow more of them and transplant them to migrate where they want to sit at the base of the hair follicle and regenerate the hair growth in multiple follicles at a time. So it's a cell transplant as opposed to a follicular transplant model. So all we're looking to do is replace the dermal sheath cup cell population in those patients with an androgen-insensitive dermal sheath cup cell population. So the fibroblasts are in the sheath of the hair follicle, and their job, as I said, is to maintain the integrity of that follicle through the expression of proteins like type 1 collagen. So we've moved two assets from the fibroblast platform into the clinic this year. The first is a phase 1/2 trial for the treatment of chronic Achilles tendinosis. It's a randomized placebo-controlled trial. We're studying VISA and VAS scores, as you saw from OrthoCell. Those are meaningful scores, but also doing ultrasound imagery. Because we think that the most meaningful image that you can get is to actually understand how much you've restored the tendon itself. And what we're doing is we're leveraging. I'll just go back for a moment. We're leveraging the clinical work of a collaborator named David Connell out of London, who did. Sorry, I'm jumping around here on you. What have I done now? I'm just pressing so many buttons. We are leveraging the work of David Connell out of London, who did 3 small phase 1 trials in different tendinopathies, including this one in chronic Achilles tendinoses, and a nice restoration of function and elimination of pain to nice statistical values against an active control. And most importantly, had this type of imagery. This is actually the image. You can see the leg bone and foot of one of the patients in that trial. It's a 63-year-old man, refractory to all other kinds of treatments, had failed physiotherapy, failed other kinds of treatment modalities, was ineligible for the trial. And this is the image below is exactly what you want to see in a healthy, functioning, pain-free tendon, is the long linear strands of type 1 collagen, which are indicative of a strong functioning tendon. This is the man's same tendon 6 months after a single injection of type 1 or of skin-derived fibroblasts. And when we were locking up our intellectual property around our fibroblasts derived from the hair follicle, we went to David Connell, who had just published this work, and we said, we really like your clinical models, really like your clinical work. We think we've got a more powerful cell type to accomplish this or better. Because we've got a cell type that's more expressive of type 1 collagen than the one you used in your trial. And he wasn't inclined to like us at first, but eventually, the long story short, has folded up shop, folded up his IP into our company, and has joined us as a clinical collaborator because we believe that the mechanism of action is just that. It's the type 1, it's the expression of type 1 collagen to affect this kind of repair that you can see in this image. So the other product that from the fibroblast cell population that we moved into the clinic in Germany this year is a clinical phase 1 clinical trial approved by the Paul Ehrlich Institute and starting to enroll patients for essentially the repair of the extracellular matrix from those people who suffer from a thinning, as we all do, from a thinning of the extracellular matrix as we age. And this is actually stained histology of young skin stained for type 1 collagen versus old skin. Not surprisingly, as you age, that extracellular matrix thins, and about $2 billion of hyaluronic acid are sold in the world per year to essentially fill that space synthetically for a period of time so that topically your skin looks more full and robust and young and less wrinkly. And what we're essentially looking to do is to inject cells that are capable of excreting the kinds of protein, not the least of which is type 1 collagen, to actually rebuild that extracellular matrix under the skin. But dermatology is rife with all kinds of fuzzy endpoints and marketing science. And what we really wanted to understand in this trial is whether we were doing something meaningful biologically under the skin. So we've designed perhaps what is one of the first cell therapy trials in the world using healthy volunteers. And this has been approved by the Paul Ehrlich. And what we've done is, while ultimately we want to treat the face and the upper necks and the hands of patients who have this, who have skin that they want to look younger, we've designed a trial for this trial, the phase 1 trial, where we're doing 4 different injections in the lower back of patients, 4 different injection points. It's placebo-controlled, but also a placebo injection point, single injection, double injection, triple injection. And then after 6 months of those injections, we actually biopsy. That's why we're doing the backs of healthy volunteers. We're biopsying those injection points to send part of those biopsy materials to histopathology to actually measure how much we've reconstructed that extracellular matrix. And then the other biopsies we're sending for FACS analysis to measure how much we've upregulated or downregulated, as the case may be, biomarkers that are as well established in the published literature associated with aging and sun-damaged skin. So what we believe we'll have, even though this is a phase 1 study, what we believe we'll have is a very instructive data package that will give us very, very informed go, no-go data on whether or not we're doing something meaningful and potentially even capable of being licensed. So the other trial that's in the product that's in clinical development is our RCH01. This is for the treatment of pattern baldness. We've completed a phase 1 trial a couple of years ago in this study, and you can see, I won't go into how it compares against standard of care. Typically at 12 months you see peak results from whether you're taking a drug called Propecia. Women can't take it. It has side effects like erectile dysfunction. A topical like Rogaine, typically you see 7 to 16% increase in hair density, but it doesn't stop the baseline progression of the disease. So eventually, an increase even 16% over 4 hairs, you don't care anymore, and so you stop taking it. What we think we're doing here is actually, as I said, regenerating hair growth with a cell population, which doesn't cure the disease, but because the cells are immune to the attack of the androgen hormone, you've got a functional cure. So the data was sufficient enough, let me say this, to be commercially meaningful to our partner now, Shiseido, which is the 4th largest cosmetic company in the world, who did a license on that technology for Asia and is co-developing this product with us now in Japan. So they did a $35 million deal signed in 2013, $4 million up front, $31 million in milestone payments, plus royalties. But the iceberg below the water on this deal is that they're also commercially, financially committed to funding all the clinical work in Japan first and in all their Asian markets, all the regulatory approvals, the manufacturing, and the sales, marketing, and distribution costs. So they've commissioned their facility, they've got PMDA approval on this, and we're moving forward, and we expect this trial to launch in Japan in the next number of weeks. So, and we have aspirations as well to do our own trial of this in Europe starting next year to really study dosing and frequency of treatment. And in the interest of time, I'll get to our final device, which is an injector device. We discovered when we were looking to deliver our cells into the scalp and skin that there was no device out there that controlled for the consistent delivery of our cells. And so we wanted to create a device that took as much of the art out of the delivery as possible. Most of the device deliveries are still up to the thumb driving the device. So what we've come up with, and this is a design-locked version of the device now that's moving into prototype build, is a device that's very elegantly designed to deliver not only consistency in the results, because that's a touchscreen there where you see our logo, but so you can program the depth and dose of delivery, but also has a number of other value-add components to it that can be used for the injection of anything you want to inject into the skin, not only our cells. So it has a number of different elements. One is this Peltier element at this U-shaped thing you see at the end of the device obviates the need for local anesthetic. So most dermal injections you see now are preceded by micro-injections of local anesthetic. That's painful because it's lots of little injections, but it's also painful because any of you have had local anesthetic in the skin. It's actually a fair amount of pain associated with that. That element actually freezes the skin on contact. The needle penetrates. It delivers the injectable as the needles retract according to the pre-specified depth and dose. So you get consistency of aesthetic results. You increase the patient experience. You better the patient experience because there's a lot less pain. And perhaps not unimportantly, you also increase the throughput that the doctors can do in an hour because you make the procedure a lot less time intensive. So just to wrap up, in 2015, we launched 2 products into the clinic. We've got the 3 products now in clinical development. We're moving our other platform, our fibroblast platform, through review in the PMDA. Because we have, again, aspirations to license not only globally, but first and foremost in Japan for all the reasons that have been talked about in a number of sessions this year. So the last slide, we're a cell therapy company, relatively early stage cell therapy company that will have 2 clinical readouts next year. Both tendon and dermatology trials will read out data next year. And the device is targeted to have a CE mark for the injection of hyaluronic acid next year as well. So all of those we expect to be licensable events. Thank you.